Hi everyone, my name is Logan and today I'll be presenting you my study on microbiome transplantation in Cuix fasciitis, a mosquito species found here in Hawaii, and its implications for mosquito fitness. So first I'd like to say thank you to Papa Jack for providing me with the opportunity to perform this research through the John and Violet Kay Fellowship. In Hawaii, the mosquito species Cuix fasciitis is a carrier of avian malaria, which is a disease that native Hawaiian forest birds are particularly susceptible to. This has caused the endangerment and even near extinction of many native Hawaiian forest birds. And you can see the decrease in population density for one native Hawaiian forest bird species in the graph on the left. However, there is a potential solution to this problem. In Hawaii, the Birds Not Mosquitoes group has been provided funding in order to perform the Wolbachia incompatible insect technique method, or Wolbachia IIT. In this method, mosquitoes are first raised in large quantities in lab and infected with a certain strain of Wolbachia, and they're all male, so male mosquitoes don't bite, they can't spread avian malaria, and they're then released into the environment to mate with wild females. These wild females have wild Wolbachia, and since the lab-raised males have an incompatible strain, when they mate, the wild females will lay eggs that don't hatch, which will decrease the population over time. However, male mosquitoes that are raised in lab have to compete with wild males to mate with the females, and research has shown that lab-raised mosquitoes have poor physical fitness under certain conditions, and they have determined that this may be due to their microbiome, as lab-raised mosquitoes gather a lab microbiome as compared to a wild microbiome. I determined that this may be able to be alleviated through the transplantation of a wild microbiome into a lab-raised mosquito, providing a lab-raised mosquito with the benefits of a wild microbiome while being raised in lab. So the microbiome is the group of microorganisms that lives in or on a mosquito or a hosta, and it's been found to affect their physical fitness. So research has also shown that mosquitoes gather a large part of their microbiome, like the bacteria, the viruses, fungi, from their larval phase, which is their first stage in the water. They then move on to the pupal phase, which is a very short metamorphic stage between the larval and adult stages, and finally into the adult stage. I wanted to infect the mosquitoes at the larval stage and then determine whether it had carryover effects on the adult stage. So in order to determine this, I asked the question, can the physical fitness of lab-raised mosquitoes be improved in Hawaii through a microbiome transplant? And I hypothesized that there would be a significant difference in both developmental rate and physical fitness after transplantation of the wild microbiome. So in order to test this, I tested 3 experimental trials with increasing breadths of the wild microbiome. The first is the mosquito midgut, which analyzes the effect of a specific part of the mosquito microbiome, then the whole mosquito, which analyzes the effect of the full larval mosquito microbiome. Finally, I tested field water, which tests the full environmental microbiome. And I tested 2 dependent variables. The first is developmental rate, which impacts how quickly mosquitoes can be raised in lab. So if mosquitoes have a shorter developmental rate, then more can be raised in a shorter period of time to be released into the environment. Then I tested physical fitness, as for the mosquitoes released into the environment, this would impact how long they can live, how they can respond to the environment, and so how they can compete with other male mosquitoes. In this project, I had 2 parts. In the first, I extracted the microbiome from their wild sources, transplanted it into the lab mosquitoes, and then tested developmental rate and physical fitness. In the second step, I sequenced the DNA from the microbiome in order to determine which bacteria were a part of that transplanted microbiome. So in the first step, in which I extracted the microbiomes, I first dissected the mosquitoes to gather midguts, and then took the sources of the microbiomes and bead bashed them or crushed them in order to make them into an easier form. And finally, I filtered those crushed formats in order to gather only the microorganisms found in the microbiome. I then took these microorganisms and transplanted them into the larval environment of the mosquito, then raised them. And at this point, from the larval to the adult mosquito, I analyzed the mosquito developmental rate. And once they were adults, I analyzed physical fitness through a cold tolerance assay. So I used cold tolerance, which included inducing a chill coma or form of sleep by subjecting mosquitoes to colder temperatures for 24 hours and analyzed how many woke up in each trial, as this is related to both ion movement and in response to environmental factors. So having these results, I asked a specific question: does microbiome transplantation improve developmental rate? And I found that the answer was yes for both the whole mosquito and field water trials. I found that the number of days that it took for the mosquito to develop in its larval phase was significantly decreased for both whole mosquito and field water trials, meaning that it would take less time for them to develop into adults and more could be raised. Then I asked, can microbiome transplantation improve physical fitness? And again, I found the answer to be yes. The field water transplant had caused a significantly increased rate of cold tolerance, or how many that were alive when they grew to, how many that were alive after the chill coma. And although the mosquito midgut decreased the amount, I found that the field water transplant can significantly improve both developmental rate and physical fitness. Knowing this, I moved on to the second part of my project, where I analyzed the bacteria in the microbiome. First, I extracted the DNA from the mosquito microbiome and amplified it. By amplifying it, I took only the bacterial DNA and amplified it, making a large enough amount to read in the sequencing that would provide me information on which bacteria were present in the microbiome. So then I took this and I analyzed the genomic data with the help of Mr. Hill using a shell script and in R script. So then I asked the question, which bacteria caused the change in physical fitness? And looking at which bacteria were found in each of the different transplants, MM being mosquito midgut, WM being whole mosquito, and FW being field water, I found that 3 bacteria were found only in the field water transplant. And so these bacteria may have caused a change in physical fitness, these being Methylophilus, Aquabacterium, Hydrogenophaga, all of which have already been found in wild mosquito microbiomes. In order to determine whether these actually caused a change in physical fitness, I created a linear graph in which I put the number of unique genera, which would be 0 for mosquito midgut, 2 for whole mosquito, and 3 for field water, the bacteria found only in those transplants, and lay them up against a cold tolerance rate, their physical fitness, and I found that there was a linear correlation. So the specific bacteria found in the microbiome did improve the physical fitness of the mosquito. I then analyzed additional properties of the microbiome, such as relative diversity, and how that could impact physical fitness. And I found that in the graph on the left, there is a positive correlation. So as the diversity of the overall microbiome improves, so does the physical fitness of the mosquito. And this includes the number of bacteria present in it, and also the different amounts of the bacteria. However, this correlation was not as strong between the number of unique bacteria in them. So I determined that the specific bacteria found in the microbiome have a stronger effect on physical fitness than the overall diversity of the microbiome. Here's some limitations of my research. So in my research, I had a smaller sample size, which allowed me to gather some conclusions, although they might have prevented me from gathering stronger conclusions or more conclusions. And I also do not know the specific properties of the bacteria that I identified to have caused a change in physical fitness. So I propose that in future research, the experiment should be repeated with a larger sample size so that more conclusions can be gathered and stronger conclusions. And I also propose that in future research, only these 3 specific bacteria should be analyzed in order to determine their effects on the mosquito microbiome. So in conclusion, my analysis allowed me to determine that the field water transplant can significantly improve both developmental rate and physical fitness. And I analyzed specific properties of the microbiome along with the bacteria that caused the change. So based on my results, I proposed that a step be added to the Wolbachia IIT method, that being that bacteria be introduced into the larval environment through either the field water method or the 3 bacteria that cause the change in physical fitness, in order to make both steps of the Wolbachia IIT method more efficient. So that would be raising mosquitoes in lab would be more efficient, as developmental rate allows more mosquitoes to be raised in a shorter period of time. And then these mosquitoes released into the environment would be more effective at competing with wild males and mating with wild females. So this improved method of mosquito control can be applied to state conservation uses, as the situation with native pine forest birds is particularly dire. Right now, the akikiki, shown on the right, has only 5 members left in the wild, and the wild population is expected to go extinct by the end of the year. So creating a more effective version of the Wolbachia IET method is very important to conserving these species. And in addition, this can be used for other species. So Wolbachia IET is also used for Anopheles, which is another mosquito species that carries malaria. So this research can also be used in order to help with global health inequalities and efforts. I had 2 main skills that helped me the most during this research project. The first is planning. I found that it was very important to have every single thing I could planned out, analyzed before I walked into the lab, so I could be as possibly prepared as I could be before going in. And I also found that organization once I was in the lab was absolutely critical. So once I was in the lab, I needed to organize myself, the lab, and especially my lab notebook. My lab notebook was when I was in the future and I looked back at my lab notebook, it allowed me to gather information that I needed from the past. And so it was very important for me to organize my lab notebook. I'd also like to provide some special thanks to some people who helped me along the way. My mentors, Ms. Waionihea and Dr. Medeiros, not only provided me with information about the mosquito microbiome and mentored me about the subjects that I needed to know, but they also inspired me to do my work with the mosquito microbiome and future work in the area, which I'm very grateful for. I'd also like to thank Mrs. Kobayashi and Dr. Chan for giving me their time to help plan my project, work it out at Iolani, and also teaching me life skills such as organization and planning and helping me in my future. I'd like to thank Mr. Hill for working with me to perform my bioinformatic analyses and for teaching me about bioinformatics, which inspired me to do further work in the area of computational biology. Thank you all for your time and help with my project. Here are my references, and thank you.